Thank you, Dr. Vesa. And finally, we have Dr. Cynthia Reinhart-King, also from Rice University.
Great, thank you. And thank you as well for the opportunity to speak. I'm going to switch gears a little bit. So I'm going to be talking about cancer mechanobiology. And compared to a lot of the things that we've talked about, mechanobiology is really just, I would say, hitting that area where it can have translational potential in cancer. And so I'm going to give you a little bit of background as to what it is and some examples of where it's headed and the impact that I think it can have. So in terms of what do we know about the mechanobiology of cancer, it's an incredibly mechanically complex system. So we now know that more aggressive tumors tend to be stiffer. We know that the cell stiffness tends to correlate in the opposite direction, which is that softer cells are often more aggressive. We know that there's a lot of compressive stresses that develop inside tumors and interstitial pressures. We know that topography changes. You get more densely packed fibers. We heard earlier about the alignment and the organization of the fibers changing. And then, of course, as cells enter the circulation, they're exposed to various fluid shear stresses. And in a lot of the areas that we already know where mechanobiology affects cells, so the vascular system, the orthopedic system, the same applies in cancer, which is a lot of what you might learn in a petri dish isn't going to apply once you start applying different types of forces and mechanical conditions. And so how do we know about all of this? Actually, the field of mechanobiology has come a long way. It started off as early as the 1920s and 30s with active or passive microbiology. And so this was basically just watching little beads jiggle inside of a cell and trying to make calculations about the viscosity of the cell. And we talked a little bit about Francis Crick earlier. This was actually his work before he started delving into molecular biology. So before discovering the structure of DNA, he was looking at the cytoplasmic viscosity of cells. And then there's really a big gap, 1920 to 1990, when we started then seeing a huge outpouring of mechanobiology techniques coming out, including optical tweezers. So instead of just passively watching beads move around, we can now move them around with light. Not only measuring the mechanical properties of cells, but measuring the forces that cells themselves are exerting using traction force microscopy. Probing the mechanical properties of cells and tissues using atomic force microscopy. Then using that information to come up with tailored biomaterials. And actually a lot of the biomaterials that we use in my lab and that we've adopted and that others have adopted as well came out a lot of the tissue engineering field. So things, for instance, a lot of Bob Langer's proteges had developed for tissue engineering we then adopted to better understand cancer. And then we started using those ideas and moving into the clinic and looking at elastography of tissues, using it to diagnose the presence of tumors, the aggressiveness of tumors. And then finally, very recently, we've been manipulating molecules at the individual molecular level using Fret tension sensors. And what this has enabled us to do is actually identify new mechanotransductive molecules, different molecules within the cell that are responding to forces so that they become therapeutic targets. And so I'm going to give you an example of how we've been able to implement some of these tools to then understand the biology of cancer. And I should have said this at the outset, but I too, like a lot of the people that have spoken, I am an engineer, but I also trained as a biologist. And the fundamental biology, I think, is what has enabled the ability to identify a lot of these therapeutic targets. So I'm going to try and show you a case example of that, looking at tumor angiogenesis. And so we heard earlier about Judah Folkman pioneering this idea of halting tumor growth to prevent, halting blood vessel growth to halt tumor growth. So in addition to that concept, there's also the opposite concept that's been pioneered by Rakesh Jain, the idea that instead of trying to stop blood vessel growth, maybe we should be trying to normalize it. And so if you look at the blood vessels within a tumor, there are often more of them and they're often tortuous and leaky. And so he's really pioneered the idea of trying to normalize them because it's shown you can improve immune cell delivery, you can improve drug delivery into tumors. The tumors, when they're no longer hypoxic, can often be less aggressive. And so how is this done? So most of the research in this field has been done looking at VEGF. So VEGF, when it was originally discovered, was actually vascular permeability factor. And so a lot of interest controlling permeability and growth by controlling VEGF. But we started thinking about it in the context of mechanics. And because of some work we had done earlier, we hypothesized that, in fact, the stiffening within tumors might actually disrupt the barrier integrity of the blood vessels within those tumors. And so we took what at the time was a fairly new system, these polyacrylamide gels. Now they're very well utilized. They're a beautiful system because you can tune the mechanics to a wide range. We put endothelial cells on them. We used a simple dye flux assay. And what we saw is that as you increase the stiffness of the surface the cells are sitting on, you normalize for the porosity of the surface, the permeability of those monolayers actually increases. And so we looked at not only the permeability, but also outgrowth. These are endothelial cell spheroids dropped into collagen, where we tuned the stiffness of collagen, and we see significantly more outgrowth when the environment is stiffer. The important part in these two assays is that we haven't manipulated anything but the stiffness of the matrix. So we didn't add in extra VEGF or any other sorts of growth factors. It's really just the stiffness here that's the only knob that we're turning, and we're seeing really different vascular phenotypes. And so this was all in vitro, so we went back and we did it in vivo as well. We used the MMTV-PYMT mouse. This spontaneously develops breast tumors. At 4 weeks, we give it a lysyl oxidase inhibitor. So this is basically, the tumors are still forming. They form pretty much at the same rate, but they're much softer. The collagen cross-linking is inhibited. And when you look at the tumors that form, you look at the vasculature, they're actually much less vascularized than our control untreated tumors. So softer tumor, less vasculature. Not only that, but the vasculature is much less leaky. So we added both a fitzy-dextran, which is the green that you see there to mark the vessels, and then we added Evans blue to see the dye leak out. In our control, our stiff tumors, we see much more leak. So again, in this situation, the knob that we're turning is collagen cross-linking, and we're getting very different vascular phenotypes. Okay, so what does this mean for translating it? How are we going to think about this? So there's been a number of groups thinking about how to alter matrix stiffness. This is actually very hard. A lot of the drugs that have headed towards clinical trials to prevent fibrosis have failed. And so our approach hasn't been to say we're going to alter matrix stiffness. We're going to alter the cell's response to the matrix stiffness. We're going to get the cells to ignore it. And in this case, we used a FAK inhibitor. And what we found is that the black bars tell you that the permeability goes up. When we apply the FAK inhibitor, we can bring it back down to baseline. So we're basically asking the cells to ignore that stiffening. And so in the time that we were doing this work, FAK targeting drugs have been moving their way through clinical trials. Defactinib is now prescribed for ovarian cancer. And we're now asking questions about whether or not we can make these drugs even more specific. And actually, so one of my postdocs, who's now a faculty member, Francois Bordelot, has discovered that stiffness doesn't just affect FAK activation, it affects FAK splicing of specific isoforms that look very specific to cancer and are much more aggressive, found in much more aggressive cancers. And so there's opportunities potentially there for splicing specific drugs. We also looked at the family of statins. So you might know statins. They're the #1 prescribed drug in the US and used for cholesterol control. They also have indirect effects on the Rho kinase pathway, which is intricately linked with cell mechanosensitivity. And so what we did in this experiment was we applied statins to our cells on varying stiffness substrates, and we found that we could bring back barrier integrity using statins. And so again, it's the most prescribed drug, but it looks like it has these pleiotropic effects that extend well beyond cholesterol. One last idea here, there's this burgeoning field of mechanometabolism, the idea that we could target metabolism clinically, and specifically in the context of mechanics. And so endothelial cells are particularly interesting. During angiogenesis, they become highly glycolytic, and the phenotype, the metabolic phenotype of this tip cell varies from the stalk cell. And so it offers this potential therapeutic opportunity because, again, it's not seen in all endothelial cells. It's seen in the ones that are forming new vessels. And so as it turns out, using a probe that tells us about the energy in the cell, the ATP to ADP, when we take our endothelial cell spheroids, we put them into collagen, These tip cells are highly glycolytic, but they're also that response, that mechanical response, is very dependent on the collagen density they're put into. So when they're in these stiff, dense collagen matrices, they are working much harder metabolically to produce these sprouts. And so it has us thinking about opportunities in mechanometabolism. The first metabolic inhibitor was approved in 2017. When I found this out, it sort of blew my mind, the idea that while so much is known about metabolism, we're now just thinking about therapies that go after it. So that was 2017. There's a couple dozen now in clinical trials, and another 5 or 6 that have been approved since then. All right, so I'm going to just take 2 seconds to talk about one other thing. Cancer mechanobiology is not a local interest, it's global. A number of years ago, the NCI commissioned an assessment of cancer research around the world. So there was a team of 9 of us that went to Europe for a week and then went to Asia for a week, visiting multiple sites, dozens of sites, and issued this report and basically said this is something people are thinking about worldwide. And now that was over 10 years ago, and what's going on since, this was a quick Google search. These are just the meetings that I could find in a quick search that are happening just this year in Europe and Asia. So this is still a really active area that's only getting bigger as people are realizing how important mechanobiology is. With that, I'll also mention biomedical engineering, the Biomedical Engineering Society, which I just stepped down as president of. It's the single biggest group gathering of biomedical engineers in the world. In 2010, the NCI approached me about creating a session. There had been no cancer sessions at that meeting. In 2010, we created a session. It got 22 submissions. Within 2 years, it became a track. So that was the very first year it was a track. Mehmet Toner and I were the first chairs of that track with 146 submissions. In 2019, it was around 116. I think it's up over 200 now. So thinking about the growth of this field, this now makes it, I think, the 3rd largest track at that meeting. So there's a lot of interest that has grown really quickly over the last decade. So really, mechanobiology field exemplifies the power of basic science in providing the foundation on which translational approaches can be built. I think that we need to understand how cells work so that we can identify these targets that we then go after. With that, I'll acknowledge our funding and the group doing this work, and we'll move to a panel.
So please, would the other presenters come and take their seats? We've been through this drill several times. Please prepare yourself to ask questions. State your name and your institute affiliation. I'm going to take this opportunity to ask a question, and it's particularly stimulated by John Cook's presentation, but I think it's relevant to much of the future of cancer research and the future of many other fields. In the Sunday paper, there was an article, this is the New York Times, that described a child with a metabolic defect who was treated with gene editing, developed through a lipid nanoparticle delivery. She was treated 3 times, edited the mutant gene, and recovered metabolism that hopefully will sustain her for the rest of her life. That therapy was delivered in a hospital. It was delivered by acquiring at the hospital level an RNA agent for the CRISPR and a lipid nanoparticle delivery system. Now, that's a hospital-based process, much like bone marrow transplant. You were suggesting in your presentation that you have a bioreactor in your hospital. You have the ability to synthesize RNA designed out of your design program to stimulate immune rejection of specific cancers. How much of our future therapeutic care is going to be designed and presented at a hospital-based delivery system versus a manufacturing small biotech or a large pharmaceutical in some centralized process. What is your thoughts there?
Thanks for that. Yes, I saw that article. It was amazing. That child was diagnosed at the time of birth with an abnormality in intermediary metabolism, urea metabolism, and those children, 50% of them die, and the others are neurologically damaged. The diagnosis was made at birth. Right away, the academic group got started on developing the therapy, and 7 months later, after that therapy had gone through cell, small animal, and even primate studies, it was delivered to the child. So really an incredible tour de force of that group, and that child is going to be much better as a result. I do think that with innovations in engineering and the innovations in mRNA therapeutics and other biologics as well, we will be able to do more of this, personalized therapies, care of genetic diseases, because it's not a good business proposition for big pharma. We will still need big pharma for large batch manufacturing, but I do believe that for these genetic disorders, for personalized therapies, we're going to see increasingly the ability through engineering to deliver personalized therapies.
Do any of the other panel members have an opinion on this?
I think technologically, I completely agree. The technology will exist to enable that. I do think we're going to need a different regulatory framework to enable that. So I think the challenge and the slowness here will not be related to the technology. It will be related to kind of the risk-benefit and kind of the regulation associated with that.
Absolutely, you're absolutely right about that. Well, there is some regulatory movement on that issue of approving platforms rather than individual personalized drugs.
Well, I'll challenge you on that. Using mRNA vaccines, oh yeah, I know, sorry, I'm going to take a few more minutes. Using mRNA vaccines for cancer research, like we heard for pancreatic cancer early on, is basically treating a patient of one. Because they sequence the tumor, they select the antigens, much like you described, and they delivered them to the patient. They didn't test them in mouse, they didn't test them in other primates, they didn't do escalating dose. They put them in the patient, and they got a response. So I think the regulatory agents, just like bone marrow transplant or liver transplant or other transplants, it might be accommodatable to this as long as the material is reproduced in a manufacturing specific way. I have no authority here, but it's just trying to make a rational argument. So I think it's something that the field needs to be aware of. There's going to be an issue of how all this personalized medicine is going to be delivered and optimized and advanced. First question, go ahead.
Yeah, hi. So this is for any of you.
Get closer to the mic so everybody.
Yeah, so this is for any of you regarding the just general immunotherapies and how individualized they are. Like there's been a lot of discussion of just, you know, you do the thing, you figure out what specific problem the person has, and then you can address that by sending it somewhere else where you manufacture their specific solution, right? So could you comment on the scalability of these sort of processes to places where you don't necessarily have the ability to immediately like ship something out? Like for instance, if you don't have great infrastructure, but there's still people being born with these various like issues.
Yeah, actually the solution that I was describing really bore out of this necessity to think about how do we actually deliver care. And I think as technology advances to the point where wireless communication, data transfer, and the ability to regulate devices like the one I described remotely become possible, then it really creates an opportunity to improve access. That was actually one of the biggest selling features of our ARPA-H grant, which the agency really appreciated, was this opportunity to increase access to a more diverse patient pool, because if you get the device and the device is providing the monitoring, then the patient can go home, and it's no longer restricted to the patient being at a hospital or even in a city which has a major medical center. They could travel back, the device is living, could be adjusted remotely, and you get real-time monitoring as to what's happening to the patient as well.
I just want to ask something that kind of keeps on that theme, but much earlier in the pipeline, right? So before we get to the dissemination, I'm curious in each of your work, what you've learned at the intersection of like social inequity. So for example, what do we know about how the, biomechanics may, how they may differ, right, in cells, in the context, right, in a host environment that's been exposed to more stress, or what do we know about, right, how immunotherapies may respond differently, and kind of what directions do we need to go if there's a lack of data to better understand those issues?
There we go. In my own work, for instance, I obviously didn't talk about it today, but we have an interest in how diet affects some of these biomechanical properties. And in fact, high sugar diets are clinically, they advise cancer patients to keep their glycemic index in check. Diabetics are higher risk for more aggressive cancers. In our own work, actually, what we've shown is that sugars can stiffen extracellular matrix, producing crosslinks, and can actually make tumors a lot more aggressive. And so when you think about lifestyle choices and equity and availability of good unprocessed foods and things like this, all of that plays into the extracellular environment in which these cells are sitting. In addition to the direct effect of sugars on cells, the effect of sugar on the matrix and the environment that the cells are sitting in is not inconsequential.
Okay, we'll take from this mic.
Yeah, sure. Hi, Craig Richard. I'm at the University of Illinois, Urbana-Champaign. And I had a question that was kind of piggybacking off of Professor Sharp's question about regulatory environments and frameworks, more about implantable devices. And there's a concern about orphan devices, like, a company produces a device and then goes out of business several years later. What happens with that? Is there a regulatory kind of framework accounting for that, or are there things in place that what you're doing, specifically Dr. Sayed, with your hammer device, that you're doing to potentially future-proof your device for these situations? We're not yet in business and hope to be soon, and hopefully we won't go out of business anytime soon. But, it's definitely an issue, when it comes to, like, in the biologics world, orphan diseases have been a good target because you can recoup, large profit margins by charging a lot to, but in the device world, that mechanism doesn't exist. If you create an orphan device that's for just a few patients, you don't really get that ability to charge what you need to recoup your costs. I think like the way we're thinking about our device is that hopefully it is broadly applicable. It can be loaded with different types of cells, and so there's different layers of product, right? So you have the device, which is the housing unit, but we also have the cells that fit in there. And the way we're kind of imagining it is that perhaps the cells are more like the software, and then the hardware is the device that's consistent and available to all patients, and then we could replace the cells for each individual product and indication needs. So we imagine perhaps a biotech industry that looks a lot different than it does today. That the drugs are produced in the patient on an as-needed basis, that there's real-time monitoring that provides a lot more information to both the patient and the clinician about that current status. So this could be an entirely new future that we just aren't really even thinking about, but as these technologies get proven out, I think it could be a much different way of how patients receive care. Thank you.
Hi, I'm Gautam again. So my question is regarding personalized cancer.
Sorry, can you speak up, please?
Hi, I'm Gautam Possibility from Biodesign Innovation Lab. So my question is related to personalized cancer vaccines. So like, what are the specific challenges and risks that are, for developing a personalized cancer mRNA vaccine, and how do you overcome those?
If I understood you correctly, you're asking about the challenges for developing a personalized cancer vaccine. Well, I think that there's a number of challenges. One is the algorithm, the pipeline for developing the amino acid sequence of that neoantigen vaccine. Right now, there's no uniformity among the algorithms. Everyone has their own approach. I think we'll learn a lot more as time goes on how to design the best neoantigen sequences. So that's one. And then the second one is the RNA modality itself. We're still learning a lot about message RNA. There are other forms of it that we can potentially use in vaccines. I mentioned the circular RNA, RNA that's more highly structured. I do think that if we're going to have personalized cancer vaccines at point of care, we'll need devices that can manufacture those for personalized therapeutics, and that's going to require not only engineering, but as you mentioned, some regulatory enlightenment to allow for that kind of thing to occur. Right now, the boxes make drug substance; they don't make drug product. For drug substance, you need to have in-process quality control. You need to be able to affirm sequence identity, purity, integrity of the drug product that patient's going to get, and you have to do that every time.